Okay, I thought I was going to get through in 15 minutes. We are so close. I apologize. So this will be part 2 of this set of slides. Just bear with me. What we should know at this point is that the amino acids, so depending on what amino acid it is, cytosine, guanine, you're going to have a long chain of those. It's going to match up with another chain, and it makes your DNA double helix. Yeah, that's what you need to know. Basically what we need to know, and this is like very specific, so this is advanced. If you want to learn it because it'll be useful for the AP test, but if you're just trying to learn as much general information, you can skip over this. There are 2 families of your nitrogenous base, which is your amino acid. Pyrimidines, which are cytosine, thymine, and uracil, and your purines, which are adenine and guanine. It just has to do with the structure, their carbon structure. That determines what it pairs up with. If we look back, we see that a purine is always going to pair up with a pyrimidine, which means that we always have that pair going on. If we look at so we have our 2 amino acids, that is our rungs of our ladder, basically. If we look at the 2 backbones or the 2 sides of our ladder, basically we have our bonds between, if we look back, so we have our amino acids in the center, we have the sugar ring, deoxyribose, and then we have our phosphate group on the very far ends. Basically, it creates a sugar phosphate unit that creates the rungs of your ladder. Things that we also should know, RNA exists as a single chain, so it's not paired up, and that's how a ribosome would read it. DNA is the 2 polymers of our amino acids, and they create a double helix, and that's what we need to know about that. Things that you don't need to know, this is more like getting very, very complicated. They run what we call 5 to 3, and then they match up opposingly. If we look actually very closely, we'll see that the phosphates are pointed down and on the right side, and the phosphates, that yellow space, are pointed up on the other side because they're actually running in opposite directions. The 5 to 3 just indicates in what direction will my ribosome read it, because it has to be read the right way. So think of it as like a sentence. It's always being read 5 to 3 or left to right, and they match up opposingly. So when they unzip, they can both be read when we are creating proteins. Yes. So we also know that adenine and thymine always pair up. It's a purine and a pyrimidine, and guanine and cytosine always pair up as well. We call them complementary base pairing. It can also occur in RNA, but we don't see, we see thymine being replaced by uracil. And so A pairs up with uracil. This is just looking at A, the far left is your DNA. It shows the amino acid joining, and the blue would be my sugar phosphate backbone. We see that we start pulling it apart, and it'll read both sides to create proteins. How does this relate to our bigger picture science stuff? The linear sequence of nucleotides in DNA is what is passed on. 2 closely related species will have more similar amino acid sequences, and we can look at that to assess how are we related. Also deals with mutations, also deals with like genetic variability, right? Gene pool stuff. All the stuff we talked about at the very beginning of the year dates back or relates back to this. This could prove really useful, this final thing. Pause this, copy it down. It gives you the monomer, the polymer, and the type of bond for the 4 main macromolecules. And I believe this is it. Go back, look at this stuff. These are 3 videos for Monday.